Yeah, because every time I look, there's like 2 or 3 more people, so I don't want anyone to miss the beginning.
Yeah, people keep trickling in. So I think the presentation should, I'm going to try to keep it pretty short.
Okay,
Hopefully we'll keep it like 25, 30 minutes.
okay,
And then the analysis portion will be the most interesting portion. So if somebody misses the
okay,
first couple minutes, hopefully it'll be okay.
okay. All right, then you know what?
Sounds good.
Feel free, go ahead and start.
Okay.
And
All right.
I'll turn me
Thank you
off.
all. What was that?
Yeah. Also, if you have a question while Nick or I are talking, the other one will be monitoring the chat. So just feel free to pop it in there and we'll do our best to respond.
Yeah, there are a lot of us. I think we're expecting a decent amount of people. So chat is probably going to be the easiest way to do questions. But yeah, we'll monitor the chat and we'll update. All right, so let's get started here. Try to pop that down there and get myself oriented. All right, so thank you all for coming. Today, we are going to be talking about different ways that we can do automated analysis pipelines with GA3 and AI. Another way to think about this is how to save yourself some time and make a computer do all of your analysis for you. The first thing that we're going to talk about today is what is GA3? Most of you have probably never heard of this before. It stands for General Analysis 3. It is Nikon's analysis tool that we use to create these automated complex analysis that we can use to help standardize some of our routines and get like really nice data from. So the first thing that we're going to look at is just some of the images that you guys gave us and different analysis tools that we can use. So the first one is pretty straightforward. We're just going to use one of our AI tools to segment out all of these little nuclei in this band of interest. Some of the other ones that you gave us, we have this image on the left looking at cells that do or don't contain some specific target of interest, as well as some of these neuronal bodies. And then we have some tools that we can use for doing 3D based analysis. And we can get some really nice images from this. And we'll talk about all of these analyses once we actually hop over into the software. But we just wanted to give you guys sort of an idea of what we're looking to do and get you thinking about what sort of ways you can use this tool and software. Some other things, this image on the left is the one that we just saw in a nice little 3D render. We can identify our cells, count them, and then look at different targets of interest within our cells, how those targets are associated with each other, and count and measure all those different features. And on the right, we can look at the area between different cells as a result of some sort of drug treatment and see how our cells are affected on more of a macro level. Real quick, before we start to get into too much information, this is just a quick screenshot that I grabbed of the analysis software as I was making one of these analyses. It's okay, we don't need to worry about everything that's going on here. The one thing that I wanted to touch on was that if you look in our analysis window here, we have a bunch of functions. These can all be adjusted, moved around. We can add things in at the beginning. If we say we forgot to do a deconvolution on our sample after we've done our thresholding, we don't have to start our image processing over. We can just drag and drop that deconvolution function in right at the top here. And it's really nice when you're first doing one analysis, you get to play with a lot of different things and figure out what works best before we make this one full automated, like one-click analysis routine. So why should you use it? The things that we're going to try to cover today are that it will save you time. We can automate all of these individual steps. You don't have to do each individual step for each individual image that you have for your entire experiment. This hopefully will save you a lot of time, even if you're doing traditional thresholding and not using some of these AI tools that we're going to talk about. One of the really nice things with GA3 is that we can standardize our analysis routines to help reduce some user error, either between users or within users. I know for me, when I was doing research, I had to do like 300 images, and I had to do manual like image measurement for each one of those 300 images. And I can tell you that in the morning when I started doing these measurements, I was way more accurate than I was at like 5 o'clock when I was just trying to finish and get out of there. So cutting out some of that error, the variability in error can be really useful. There won't be zero error in these images, but it will be constant and repeatable. which can be really useful when we're trying to measure some of the data that we're looking at. The third thing we're going to touch on is that we can batch run these analyses. There's a tool within the software that I will show you. Once we have one of these full analysis files ready, we can point it at a folder with hundreds of images and run the same analysis on all those images without having to do it one step at a time. And the final thing that we're going to talk about is the AI tools. These are a couple of tools that we can use to do things that are normally very time-intensive manual processes, thresholding out difficult to threshold images. We have some pre-processing and some transformations that we'll talk about. So let's get into the AI tools first. Lauren, do we have any questions so far?
Nope.
Nope. Perfect. All right. So NIS AI is what we call our suite of AI tools. If you've been using the software at all in the last couple of weeks, I know that it's been at the core for about a week or two, but I know that some of you guys out there already have some of these tools. So we're just going to go over sort of what they all do and when you might want to use them. There are two main kinds. The first that we're going to talk about are pre-trained AIs. These are all ready to go. You can click on them and use them anytime you want. And we mainly use these for like pre-processing. The second kind are going to be trainable. These are going to all involve some sort of user input where we have to train a specific network on our individual samples. So my convert may look different than Lauren's, and our segment objects are probably going to work on different things, which is really great because we can train multiple different neural nets on different objects and get different and accurate results. So the first one we're going to talk about is denoise. This is probably the AI tool that I use the most out of any of them. You can see on the left, we have our standard wide field or confocal image, whatever we have. It looks a little bit hazy. We have a lot of background noise and shot noise from the camera here. And what that's going to do is it's been trained on like thousands of images, I think, to just learn what noise is and remove it from our images. So we get these nice contrasty images on the right here that are really good for doing analysis, and they'll help us with our segmentation and our thresholding. Again, this is one of the ones that I use on pretty much every image that I take. It's just like, it's a good starting point, and I highly recommend it. The next one we're going to talk about is Clarify. Clarify is going to work really, really well on thick Z-stacked sections for wide field images generally. The image on the left would be like a standard wide field image for something that we do a lot of Z-stacks on. You'll recognize if you've taken these sort of images that they just get really hazy because of all the out of focus light that we're capturing through the Z-stack. So what Clarify is going to do is it's going to give us sort of like confocally like pictures. And it's going to cut out all of that out of focus light and just give us a really nice in focus image here. It is going to behave sort of like a deconvolution, but the difference between a deconvolution and what Clarify is doing is that deconvolution is going to reassign our pixels or our light back into the point of origin based on the point spread function, where Clarify is just going to chop out that out of focus light. And as a result of that, it is not quantitative if we're looking at trying to measure like intensity based measurements, but it does make very nice pictures. The next one we're going to talk about is the restore function. If you think that this looks kind of similar to the Clarify one we just saw, that's because it is running a combination Clarify, denoise, and a deconvolution. This was made to give you the highest publication quality image that it can. This one we don't really use that often, or I don't use it very often. The main purpose for this would be if I was going to like take a really nice picture and I want to submit it to an image competition or put something on the cover of a journal, maybe I would pick a few images that look really good, run a restore AI on these, and then that's what I would like submit or publish. So a specific use case for this, but it does make very nice looking images. Those are it for the pre-trained ones that you'll probably use. So now we're going to get into the trainable ones. Like I said, these are all going to involve some user input. We're going to have to train these ourselves with images that we generate. So the first one is enhance. That's going to take an image on the left here where it's going to be either really noisy or we're not going to get a lot of good signal from our original image, whether that it just doesn't produce a lot of light or we don't want to expose our sample to a lot of light. Maybe it photo bleaches really easily. And we can train it to give us a really nice high contrast image that we can then use to segment out much more easily. This is a really cool one, and there are a lot of instances that we can use it on. It does require that we take a standard image, this like low quality noisy image, and then we do have to find a way to give it a good high quality image to train it on that it can then look at. This is sort of like kind of like training a denoise, but it doesn't function quite the same way, and we can use it on our individual samples. The next one is going to be convert. This one can be a little bit confusing, but you guys actually sent us one of the data sets that we trained convert on, and it looks really cool. So it will create a fake channel from a standard channel that we give it. The most common use case that we see for this is for a DIC image that we want to have a DAPI channel for. Sometimes we don't always want to hit our cells with like damaging UV light to get our DAPI image. So we feed it one image where it has a DIC image and a DAPI stain, and we train it to give us a DAPI image from the DIC image. And it does a really good job at this, and we can use that to count our cells, threshold them out, measure some areas, things like that, from this sort of AI-based convert channel. The next one we're going to talk about is segment. Segment and segment objects, you may have noticed, are two functions that are somewhat similar. So I'm mainly going to talk about the difference between the two of them. Segment is going to be really good for any segmentation that you're going to do, except for things that are small, mostly circular, and probably grouped closely together. That's when I would use segment objects. But segment in this example, you can see we have like a DIC picture of some neuronal cells that have these dendrites, and we want to train the AI to segment those dendrites out for us. This is a really powerful tool, and it can do tons of segmentation, and it can do it very accurately for us. The next one, like I said, segment objects. This is mainly going to be used for nuclei segmentation. That's probably the main use case. Or like you can see here, we can segment out these sort of nuclei shapes from this GFP image. The main difference between segment and segment objects is that segment objects is going to do a really good job at separating out things like multinucleated cells, nuclei that are really close together, that are crowded. It'll separate all those out and it'll give us much more accurate segmentation than you might traditionally be able to without going in and just manually counting all of your cells. And we have a really good example of this one too, so you can kind of get an idea of how it works. Next, you might be wondering, how does the AI actually do what it's doing? We're going to touch on that a little bit. You could probably fill like an entire graduate degree with what AI is actually doing. So we're just going to give kind of a short intro in the context of what the AIs are doing for our AI tools specifically. So the first thing that we should talk about is how the AI is actually viewing our images and what it's trying to do when it's creating these binaries and new images for us. So the image that you're seeing here is probably some sort of coral polyp. And this is the image that we would see if we were looking under the eyepieces of a microscope. The second that we take a picture of this, we're taking all these photons and we're going to convert them into electrons. And that's going to be read from our camera. And that is going to produce an image that looks a lot like this. This is a bit of an exaggeration just to sort of show what the pixels are doing. But this is the image that you will see on your computer screen. What this image actually is and what the AI is seeing is just an array of numbers that represent some intensity value from this image. We don't need to be too concerned about what all these numbers are and what they mean. But when the AI is trying to decide which things to include in our binary or how to make these pictures for us, it's going to do a pixel by pixel analysis of each of these pixels and come up with our final result. And we'll talk a little bit more about kind of how that actually works later, but I wanted to get this idea across first. What is a neural net? A neural net is probably a term that you're familiar with in the context of AI in general. A neural net is, if you look at the right picture that we have here, a net made up of a number of nodes that are all interconnected in multiple layers. Without getting into too much detail, that is the structure of the neural net. Each of these nodes is not dissimilar from a neuron in function, hence the name neural net. We have a bunch of inputs that we pass through our cell body, our node. These inputs are going to be pixel information, and it's going to pass through the body of our node, and that node is going to be some mathematical function that is going to manipulate our data in some way. That new data is then going to pass through the axons of our node onto the next layer. And what this functionally looks like on the right is that we have our input layer, which in our case is going to be some image that we give it. It's going to go through all of the different hidden layers of the image, which is going to be where all these functions live. And it's where the AI is going to decide what is and isn't included. And that's where it's going to be making its decisions. And then finally, there's the output layer, which gives us the final result. What that is going to functionally look like for us is something like this. We don't need to worry too much about what each one of these layers is doing. I just want to note that for something like the segment objects that we're using, there's probably going to be 24 or so layers with a bunch of nodes, and we might have thousands of connections between these. And each of those connections is going to have a weight assigned to it. And the weight is what is going to tell the neural network how to do the segmentation. It's how we can say, get different outputs from the same net, we assign different weights to it. So for this example that we have on the screen, we have our input image, which is some image of a street with some cars, and we pass it through our network, and our output is going to be some binary segmentation where we segment out, say, the roads, the cars, the trees, the sidewalk, that sort of thing. In order to get this final result, we have to train the network. How we train the network is we give it an input image, and we give it a ground truth binary, which is the binary that we want to train the AI to make for us. What it will do is it will randomly assign all of the weights in the neural network. It will take our input image and it will pass it through the network. It will spit out some binary for us. It will then take that binary, it will compare it to the ground truth binary we gave it, and it will ask itself, how far off am I? For example, the first time it runs through with random weights, it might be 25 to 35% off, something along those lines. So it'll say, okay, I didn't do a very good job. Let me try again. It will change the weights in our neural network, and it will run through again. The second time it does it, it might only be 20% wrong. So it says, okay, I'm doing a little bit better. And it will continue that process again and again and again until it approaches some value that is getting very close to what we actually want. And what that functionally looks like is something we call the loss function. This is a screenshot taken directly from the software, and this red line is what we're going to be looking at right now. You can see we're measuring the training loss over the number of iterations that we're training this neural network on. You can see at the beginning we have a very high training loss. It will quickly decrease, and then it will slowly sort of taper off to some sort of minimum value. And this value is what we are using to measure how accurate our AI is. There's no right or wrong value for what our training loss should be. Generally, a number of 0.05 is what we're shooting for. In this case, you can see we got down to 0.01. So this training did very well. But this is how we're going to measure how well our AI is doing. And this is a common way to measure pretty much the success of any neural network. All right. Any questions about AI-based stuff? No? Awesome.
Things popped up.
Cool. Okay, so one important thing to remember here is that the AI is only going to be as good as the ground truth binary, the training binary that we give it. if we have a training loss of, let's say in this case, 0.01, which is very good, if we don't give it a good ground truth binary to start with, it's not going to give us accurate data that we actually care about. But it will give us a good representation of the bad training, if that makes sense. So it's important to think about when we're capturing these images, making sure that we actually give it good data to start with. Good images in is going to give us good data out. That's a pretty good way to think about things. So we're going to talk very briefly about things to look for in our images. A lot of this is going to be familiar to pretty much anybody that's done any microscopy or any imaging of any kind. But there's a couple things to think about, especially when we're thinking about what sort of data we want to feed to our GA3 and our AI. That's just going to be important to think about. So the first thing we're going to talk about is resolution. This one's pretty straightforward. We have an image here of some structural little cavities. Our bottom right image is going to be our highest resolution image, and our top left image is going to be our lowest resolution image. For this, we're going to say that we want to measure these small cavities within our image. And if we look at the bottom right image, this looks really great. We'd really easily be able to segment these objects out, measure the area, do all sorts of stuff. As we move our bottom row from right to left, we can see the middle image still pretty good. We could still segment these out, and I think that we could get good data from them. As we get to this bottom left image here, it's going to start to become difficult to get an accurate measure of how big these cavities are, just because we're losing some of the resolution on these walls. And as we go to our top right image, we're really not going to get any good information from this. So making sure that we can actually measure the structures of interest is going to be critical when we think about how we want to measure these. Ways to improve resolution. Aside from point scanning confocal, where we can change the size of our pixel and our zoom, we're going to just want to go up in magnification, making sure that we can see our features of interest, go up in magnification, and maybe do some stitching if we need to. The only other way to do that would be to look at the camera that we're using. Camera technology has come a long way in the past couple of years with the invention of like CMOS cameras and cameras that have much smaller pixels. So we can get some improvements there as well. The next thing we're going to talk about is exposure. Exposure can pose a lot of challenges for us, specifically in the underexposure side of things on the left. If we look here, we can kind of see that we have some cell bodies here, but they're going to be really difficult to segment out because they're just so close to the noise floor and the background in the image here. This may be a good example of where we would use like an enhance AI, but if we want to do intensity-based measurements, we're probably going to have to find some way to get better signal out of these. Slightly less common is overexposure, but still something that pops up and we have to think about. Everything that's glowing pink here is going to be overexposed, and we're not really going to be able to get good nuanced data out of this. The best way to think of overexposure is if we're climbing a mountain and we have some cloud layer. If our cloud layer is above the top of the mountain, we can always see the entire mountain, how far up we have to go. As that cloud layer starts to get below the peak, we stop being able to see all those details at the top, and we don't know what's going on up there. And that's the best way to think about overexposure, is that we don't actually know the true information here. All we know is that it's very, very bright. Best way to consider and think about exposure, ways to adjust and account for it, is making sure that we have good exposure in our image. For underexposure, usually that means taking longer images. And for overexposure, usually that means taking shorter exposures. The only other way that we can really account for this is, again, with camera technology. There are cameras that have really high dynamic ranges, so we can capture really bright and really dark pixels at the same time. There are cameras that have much higher quantum efficiency, so they're much more sensitive, and different camera technologies that we may need in order to pull the data out. That's not to say that we can't ever get data from images that aren't perfect. We certainly can, but just things to think about. And the last one is focus. This one's pretty much the most straightforward, but worth mentioning. The image on the left is of some nuclei that we're interested in. These are nicely in focus. We can pretty much point out each individual nuclei, and we'd be able to measure these and get whatever information we need about them. The image on the left is from actually the same sample set as the image on the image from the right is on from the same sample set as the image on the left. If I didn't know these were nuclei, I would have a really hard time picking out any of these individual features. So just making sure that our images are in focus is going to really help us when we're trying to make a good GA3 recipe that's able to get good information from all of our images. Ways to double check focus are make sure that all your images are in focus before you image. And if we have some images that are really thick, that maybe some parts are in focus and some parts are out of focus, usually the answer here is just to run a Z-stack of sorts. So now that we have sort of a good idea of what AI is, what the GA3 is, and what kind of data we want to give this stuff, we're going to talk a little bit about some basic image processing. And the first thing we're going to discuss here is binaries. Binaries is going to be the main way that we threshold our images and we use binaries to get information about our targets of interest. So binary operations, often we'll have multiple binaries in an image. Usually we'll have, one for all of our different channels, one for DAPI, one for FITSI, one for TRITSI, and we'll want to know how those channels interact with one another. So the way that we do that is through binary operations. There's a couple binary operations. I'm going to go through them pretty quickly, but I just wanted us to have an idea of what we're looking at so that when we see these things in the software, we kind of understand what's going on. So here we have two binaries. We have our A binary and our B binary. These are two circles that overlap with each other. The first operation we're going to talk about is AND. The AND operation is going to give us a binary that is just the intersection of our input binaries. So pretty straightforward. The AND operation is one that we use all the time. The OR binary is going to be the binary that includes A or B or both. So that will include everything that we have in either one of our input binaries. The next one, XOR. This one, sometimes people get confused with the previous one. It's very similar, but XOR is only going to give us a binary that includes only A or only B and not the intersection. Equivalent is the opposite of XOR. It will give us only the intersection and everything else. Subtract. A minus B will give us the binary of A minus any part that it intersects with B. This is one where the order that you do it in matters. So B minus A will give us the opposite of this. For the next couple examples, we're going to talk about, I've added some little squiggly sections to each of our binaries just to give us a little bit more context. And you'll see why that matters in just a second. So the next one we're going to talk about is having. A having B will return full objects in A only if they interact with a B object. And another, like a way to help us think about that is the B having A. This is the opposite of A having B. Again, the order matters here. And then we also have A not having B, and this is going to be similar, but it's only going to give us objects where they don't interact with the B binary. And B not having A is the opposite of that. So those are the binary operations, and you'll see a bunch of examples of those once we actually get into the software. The next thing we're going to talk about very quickly is processing. We often want to process these binaries to help manipulate them and get a little bit better representation of our data. So on the left here, we have our original image. It's kind of like a letter 3 shape with some little appendages coming out of the back of it. What an erosion will do is, well, there are a lot of binary processes, probably like a couple dozen. So it wouldn't make sense to go over all of them right now. And a lot of them you might never even use. So we're going to go over a basic 3 or 4 here. And a lot of the processes are built off of these. So it'll help us just get a base level understanding of what we're doing when we hop into the software. The first one is going to be erosion. Erosion is going to have an eroding effect. Essentially, it's just going to reduce the outermost pixels from the image. So every pixel that touches a background, it will just turn into a background. And dilation is going to be the opposite. Every pixel that touches a background, it will make that background pixel part of the object. Pretty straightforward. The next one is going to be open. Open is actually just a combination. It's an erosion and then a dilation. It's really good at removing like tiny filaments and small objects from our binary. You can see that we maintain the original shape of this 3, but we got rid of those little appendages off the back. And a close is just the opposite of that. It's a dilation followed by an erosion. And this is a good way to remove little filaments of background in our object. So you can see, again, we maintained that same shape. We maintained our object appendages, but we just got rid of those little black parts of the 3 there. Questions about any of that stuff, basics of image analysis. We'll go over this a lot more. So if it doesn't make perfect sense, it will probably make more sense once we hop into software. Cool. All right, so now we're going to get into probably some of the most common questions that we get whenever we do a GA3 sort of presentation workshop. And the first one is, how do I build a GA3? We saw that window before. There was a lot of functions in there that were all connected and didn't really know. It seems overwhelming, and it can be at first. But usually when we're building these things, I try to break it down into like 3 main sections. The first is going to be, we're going to do all of our pre-processing. That's going to be where we do our denoises, our deconvolutions, our enhances, our converts, and we sort of decide which channels we want to use. From there, we're going to do all of our binary creation. This is where we're going to do our segment objects AI, do our standard thresholding, and then modify these binaries with our operations and our processes in order to get the data that we want from it. From there, once we have all our binaries and we've decided which data we want to use, we're actually going to do our data management section. I haven't really talked about this yet in the context of GA3 because it's not very exciting to put in a PowerPoint. But this is where we decide what images, binaries, and graphs we want to save. We can get all of our data into nice tables. We can do statistical analysis, t-tests, ANOVAs. And we can make graphs, bar charts, scatter plots, get our error bars, and decide which of those data we want to actually save out. So we'll talk about this more, but just sort of when you're thinking about how to build these, I usually try to break it down into a little bit more manageable chunks. The next most common question we get is, I don't really know what I'm doing. Like, where should I look for help? The first place to look is usually the help file. We'll show you guys how to get that. There's a lot of places to access the help file. The help file is going to, for any function that you see in GA3, you can right click on it and just go to the help file, and it will take you exactly to where that function lives. It will give you a short description of what it does. And it's really useful. I think when I was building out these GA3s, I probably used the help file like a couple dozen times just because I wasn't sure what everything does. So this is one that I use all the time. It's really useful. And there's tons of stuff in here, not just on image analysis, but on, plenty of other things as well. The other place to look is the Nikon e-learning site. This is a free website that anybody can use. It does require going onto the website, and you have to submit a request to get a login. But on it, you can find tons of instructionals for how to make different GA3s, how you would use different GA3 functions. And there's also some basics on just basic image analysis as well. So this is a really good place. This is where I would point somebody who doesn't really have a strong background in image analysis or Nikon the Element software in general. This is a good place to just go through some of these courses, get a better idea of what you're working on. And there's some more examples of more standard basic analysis in here too. Okay, that was a presentation. We did it. Congrats, you're all experts in GA3 and image processing. If anybody has any questions, now's a good time. We'll take a quick 5-minute break so I can swap over to my desktop computer. And if anybody has to go to the bathroom and grab a snack, now's a good time to do all that stuff. All right, let's see.
Sorry. Can you hear me?
Yes.
I can, yes.
Hi. Sorry if I missed this in the beginning. I joined a little bit later.
Yeah, sure.
Will you be doing some demonstration using some of the data sets?
Yes, yeah. We're going to hop over into the software right now, actually.
Thanks so much.
So I'm going to transfer over. All right. Camera change.
So the restore function, I'm going to answer the pop-up question. Restore AI is just like Clarify, it is not quantitative. What it's doing is it's looking at your image that you've acquired, and it's going to make judgments based on that specific image. If you ran restore on 2 images, it's going to make different judgments on those 2 images. So you would never be able to make a direct comparison between those 2. It's doing a combination of denoising, decon, some declarify. It's kind of like a little magic wand that makes things look really pretty, but I wouldn't say that it would be consistent between every image that you're taking. So it's mostly to say, this is the prettiest I can make this picture look to display it, but I wouldn't do any sort of quantification or analysis on a restore AI image.
So along those lines, how are journal editors and reviewers reacting to modified images with enhance and convert and are you touched on quantitative analysis and how that might be affected, but how is the data considered rigorous, and how's that coming along?
So I wouldn't say that we personally get a lot of feedback from journal articles on our end. The AI algorithm that we have produced that we know is quantitative, both pre and post processing, is denoised. We have a white paper that we've published for that so that you could submit it to whoever you're doing your submissions to. With Clarify and Restore, they're trying to get more conflictuality out of an image that you took in a wide field. I personally highly recommend that you report every single thing you do to every journal article that you're putting in. One of the nice things about these GA3 recipes, and Nick will show you how to do this in the software, you can save them out and submit them when you're doing a paper submission and say, this is exactly the recipe that we used on these images, just to kind of give a more structured analysis flow for whatever paper that you're submitting to.
Yeah, I would think you'd have to disclose that you used AI to process the image, and this is how you did it. So are you seeing that or have you looked at publications where people are sort of doing that?
That was why we built in the save export functions so that people could export both the AI algorithms. So you can get a specific AI file for any of the ones that you've produced. You can share them with colleagues or with anyone who wants to replicate your data or try and see about what you did. And you can save out GA3s. So they are designed to be shared between groups and 2 papers. So
Thanks.
And Nick and Lauren, I apologize. I have another meeting that's a conflict that's coming up. So if it's possible to start with the Retinal ganglion cell images, that would be great.
which one were those? We kind of numbered things.
That is the
Is that your 5?
that is the last one, unfortunately. Okay. We can start with that.
That would be great because I have to leave in probably 10 minutes. I apologize.
All right.
We will say we kind of organized it a little bit with some features that I talk about, and then Nick talks about. Oh, well, Lauren talked about this earlier, so it might not flow as well as we did before, but yeah.
All right, so let's do that one very quickly. Again, we're going to be a little bit out of order here, but in order to try to get that done, let's go for this one. So let me share this. Oh, this is not going to, it's going to get mad at me if I try to share. All right, 1 second. I am on my desktop here. Can I let you share? Oh boy. All right. We may have a technical difficulty here. Can I change it so that I can actually let myself share this? Sorry, everybody. I'm had to switch to my desktop because it's a better computer for running analysis. And I somehow set it so that only certain people can share. Let me pull up the meeting options. who can share, who can present, everyone, save. There we go.
We do recommend you do these on a desktop that has a decent processor in it.
Yes. All right, can everybody see that?
Yes.
Perfect. All right, so I'm gonna switch my mouse. So this is what this analysis is going to look like. A caveat kind of before we start is that none of these analyses are going to be fully baked. I think the main goal for this section was to sort of go over common problems that we might run into, common ways that we would solve them, and give you some things to think about when you're kind of making these analyses for yourself, and kind of give you just a good idea of how to navigate the software to make it a little bit easier for yourself. So before we get too heavy into this, this is going to be the layout for anybody that's not familiar with elements. I like to try and keep things pretty organized. So I have all the tools that I might need over on the right side here, and everything else I kind of try to leave open. For this analysis, for everybody who might not be aware, this image on the left, we have 3 channels. We have a DAPI channel, a FTSE channel with some neuronal bodies here, as well as a Psi3 channel. We wanted to do 2 main sorts of analysis here. The first kind is going to be figuring out which nuclei are associated with these like bright red channels here. See. So generally when I'm going to make one of these recipes over here on the right side, I'll zoom in a little bit so that we can see a little bit more clearly. I'm generally going to start up here with my image processing. And I'm going to work more or less left to right, top to bottom. You can see here in our image processing, we have all of our deconvolutions, our background adjustments, our contrasts, our denoising, that sort of thing. In elements, we try to name things very similarly to what you would expect in like other image analysis software like Fiji. So your erodes and dilations are going to be the same. You know, we don't call it deconvolution some other weird name. So if you have an idea of the sort of analysis you want to do, you should be able to find things pretty easily. The other thing to note is that we can scroll, we can zoom in, we can focus on things. You'll notice I can make little sections here so that all my functions sort of live in one box that I can navigate more easily. Also, as you can see, I've tried to break these down into those kind of 3 main sections we talked about, where I do all my pre-processing and then I work on my binary layers and get all the information I want from those. And then at the bottom, I sort of do all my records and get all my tables together and any graphs that I want. When you open up a file, you're generally going to want to turn your preview on. What that's going to do is it's going to make it so that you can see all of your channels live. We have these little, see if I can zoom in even more. We have these little like black chiclets, and every time one of those is activated, that will show up live in our image you can see over here. So at the top, we have our inputs. This is the image that we start with. It's our red, green, blue image. And I like to generally work one channel at a time because it makes it a little bit easier for me to see things. So for this one, the goal was to identify cells that contain this red target. And the first thing that I did was I played around a little bit with some of the pre-processing. There's a lot of background in this image. In our darker sections, you can see, even though it looks sort of black here because we've have our lookup tables on, we're getting pretty high counts. Like we're getting 150, 100 something counts. And the first time that I went to threshold these out, I was noticing that I was picking up a lot of stuff in the middle, but not a lot of stuff at the edge. So I ran an auto shading correction. And to find and sort of navigate these functions, you can right click on the white section and it'll pop out this menu. If we zoom in a little bit, oh, you can also, I should have taken that out. I'll show that in a second. You can locate any function by hitting the locate tab, and it will take you to where that function lives. in the like interface on the right side here. The other thing that we should note real quick is that you can see we have all these connections between all of these things. If you click on any function, it will show you what it's connected to and what is connected to it, which can help us navigate around our GA3 a little bit. And the other thing to look out for is some things like this threshold that I'm doing. When I drag it in for the first time, it'll be all grayed out. And you can see that it's looking for a channel. So I won't be able to connect this to like a binary, say. I have to connect it to a channel image. And the way to differentiate those is that channels will have solid colors and binary layers will have this little symbol, and they'll be colored the color of the binary that they represent. And at the bottom, you can see tables have this little table graphic, and charts will have a chart graphic on there. So that's how to differentiate what nodes are doing what. So like I said, the first thing that I ran on this is an auto correction or auto shading correction. And what that's going to do is, if we look at it live, it's going to give us a nice even illumination of the whole field. And it's going to make thresholding this out a lot easier. From there, what I did is, because I saw that we have a ton of background here that we might not be interested in, I thought that it would be a good idea to do a background subtraction. So I will look up table here. The way that a subtract background function is going to work, this is a pretty common one, is we first need to point it at an image, and then we need to give it some sort of threshold that we've come up with. So if we look at that threshold, anything that has these three little dots on it is something that can be double clicked on, and it will open up the options for that. So here I thresholded out the background, or what I thought looked like the background. And you can adjust all those settings here. You can see that we can change those, something like that. And I did a background subtraction, and that, let me zoom out again.
Nick, I would zoom out. You're rendering a little weird in the share.
Oh, okay.
There you go. Yeah.
All right, that's better. Yeah, sorry. Let me know if you can't see something. We're trying to, we know it's like things are small, and so I'm trying to zoom in so that we can see what we're doing. So I ran a background subtraction on this. It is important to note, this is sort of like what I thought was going to work well. If you guys, who are the ones actually doing these analysis, we don't really want to make decisions for you on your science. So some things that I excluded here, you might say, no, I actually want to include that in your data. So I would go in and mess with these recipes and adjust the thresholding, things like that. Maybe try something else that you're used to doing. So this is what we thought would work well. Like auto shading corrections and background subtractions are pretty common pre-processing routines. So we kind of just wanted to show like where you can find those, and how to access them and drag and drop them into a GA3 if you were the one to make it. So now that we have our nicely sort of processed image, I'm going to want to run a threshold on this. I ran for this, you can see I ran a threshold. It's connected to this background layer. You can connect it to like the traditional layers, but it's not going to give you as good results. And this is the threshold that I came up with. Another quick shortcut that you can use, if you hold Control and hit the up arrow key, you can change the opacity of these binaries, which can be useful if you're trying to see what you're doing. I usually keep them like 20% opacity, just so that I can get a better idea. And again, this is what I thought were the cells of interest. And because we want to look at an intersection, I did one of the erosions that we had mentioned earlier. And that's just going to shrink the binary to try and avoid getting any like false positives from things that might just overlap a little tiny bit. So this binary looks good for me in the Psi3 channel. So the next thing that I did is I'm going to get rid of all that stuff, and I'm going to go into my DABI channel. We're going to not look at this one for now. This is a segment objects AI. There are some pre-trained AIs that just come with the software. I don't recommend using them for a ton of stuff. But when I was kind of messing around with this one, I just tried one of the pre-trained ones, and it actually worked pretty well. You can see in some places, like here, it didn't quite perfectly capture that. It has some fragments and things. But for what we're trying to do here, where we're trying to find the overlap between these two binaries, it worked really well. This might be something where I might try to do a traditional threshold, but this looks pretty good. So I opted to keep this one. And then same thing as the other one. I did an erosion here because I wanted to try and get rid of any false positives. So that's just going to shrink everything down. The next thing I did for this one, we have a function called remove border objects. What that's going to do is get rid of any object that it detects as touching the border. What that looks like is if we activate that, you can see it gets rid of a lot of these half objects on the periphery. This is really common when we're looking at like fields of big cells, because those half cells are going to throw off our measurements if we're trying to do like areas or something like that. We might not need it for this one, but I like to put them in just because I find that it's really helpful and it's not going to throw me any like weird data that's going to throw off my averages. If you want to know where that lives, we can go look at that as well. And that's going to live in binary processing under our touching borders, removes objects touching borders down here. And the final thing I did on this one, when I ran this erosion, I noticed that I had some sort of like little objects here that I didn't really want in my image. So I did a clean. Clean is going to be one of the binary processes that's going to live in up here at the top. It's one of the first ones. What clean is going to do is it's going to, let me see if I can find a good example. It's going to remove any of these tiny objects of a specified size. And I did that just to, again, clean it up a little bit and get rid of any false positives and false negatives that we might run into. So from there, we have our two binary layers. I'll zoom in a little bit. These colors don't contrast great, but I have two more where I'm going to actually get the data that we want. And down here, like we talked about, we did all of our binary processing, and now we're going to work in our binary operations. So the first one that I'm going to do here is the halving binary. You'll notice that I have nuclei having psi3. And when I activate that binary, you can see if we activate, let's activate this channel as well. You can see that it does a pretty good job of only showing me the nuclei that also have this psi3 red channel in them. I believe I went and talked to this group to sort of go over some of their data a little bit. And this, I believe, was the one thing that they wanted to do. But when I was making this initially, I also put in the not having binary so we can see how it's doing with identifying cells that don't contain this psi3. And I think for the most part, it does a pretty good job. So this is a pretty straightforward little GA3 recipe that we can make to really easily identify cells that do or don't have some feature of interest. And this would work really well for kind of any analysis that you want to do that's sort of related to that. From there, I moved over into our second part of the analysis, which is looking at this FTSE channel where we have our markers of interest. So I'm going to turn off all of the things that I don't want to look at for now. We're going to zoom out a little bit just to get an idea of what's going on. And make this window a little bit bigger. Here you can see we also had a lot of background here. So I did the same thing that I did with the psi3 channel. I did an auto shading to help bring out and get me a little bit more even illumination to help me image it. I did my background threshold, which in this case was a lot of stuff. And then I did my background subtraction from that. And if we look up table this is going to give us a much easier to work with image where we can see these neuronal bodies a little bit better. And from there, the information we wanted from this was the size of the cell body itself. as well as information on the length of the dendrites coming out of them and how many branches are in each dendrite. So the way that I did that for this particular one, the first one I did is I just did a standard threshold for these soma bodies. We'll increase the opacity there. These images are pretty overexposed, so I'm not sure exactly what's going on here and the exact area that we're interested in. But because they were a little bit overexposed, they were pretty easy to threshold out. And that is a pretty straightforward threshold. I thresholded out the bodies. I did do a size exclusion just to get rid of any of the little ones in here. If you guys wanted to include those, you can always manipulate this data to include stuff, not include stuff. Yeah, that's set at 16. So that one was pretty straightforward. From there, we wanted to measure the dendrites themselves. So I ran a separate threshold off of the same channel, just to try and capture as much of these dendrites as I could. From there, I used a very common binary process called skeletonize. We will probably need to zoom in to see what this is doing. Skeletonize lives down in the skeleton section of our binary processing. What skeletonize is going to do is make a single line pixel binary from whatever threshold we give it. So you can see here, it does a pretty good job. We get some sort of little segments here that are not exactly what we want. This is something that I would probably highly recommend using the segment AI on. If I could go in and hand draw these sort of neuronal connections and train a segment on it, and I think it would do a really good job, even if we have like, not perfect image quality. So that's a really good use case here. I didn't personally train one in this case because I wasn't sure like what exactly would be of interest to this group. For instance, like I don't know if this one would go all the way out here. And like I said, we're not really the ones to make decisions on your data. So I would, I'm happy to work with this group and sort of help figure this out. So I ran a skeletonize, and I'm going to use this to measure the sort of branching of these dendrites. From there, you can see that in this one, like I picked up some really small objects here that I don't really want. So I used this function. It's called filter objects. It lives also in binary processing down here. This is a really common one, and this tool is super useful. I use this all the time. In this instance, I just calculated any length that's greater than 25, I want to keep, and it will get rid of anything that's less than 25. So when I activate this function, you can see I get rid of that because I'm pretty sure that this isn't something that I actually want to measure. And you can filter based off of tons of different things. So there's a bunch of different object size filters, shape filters, position, and intensity-based filters. This is a really good tool for almost any analysis that you can use to sort of get rid of some of the stuff you don't want if you're not able to perfectly threshold it. The next one you'll see here is color by ID. I talk about this actually in the other analysis that I'm probably going to do. But this is another really popular function and one that I use a lot. It's not going to look perfect here. I just like to use it for checking. It will change every object to a different color. And that's really nice to see like which objects are connected and which ones aren't. Obviously, this is a green. It changes this to green, which is really difficult to see. So I don't personally like it here. But over here, you can see that we have our different objects. So I'll keep this one on for now. And then within this, one of the things we wanted to know is how many branches are in each of these dendrites. And that's where I'm going to use, we have a function already built for this. It's called detect branching. It's going to live in the skeleton section of our binary processing. What that's going to do is if we zoom in, if we activate this, you'll notice that it creates a single pixel at every point that it detects a branch. And this is really, really useful tool, especially for this. It's going to do exactly what we want. And in order to measure how many branches are in each object, we're going to use what's called a parent measurement. This is something that Lauren will talk about in more detail. This is part of the parent-child relationship measurements that we use. We use them in lots of different analyses to measure things. And you'll notice that it is connected here. So what parent is going to require is it's going to have us give a parent, which in this case is going to be the entire dendrite, and it's going to have us give a child, which in this case is going to be the branching. So what this will do is if we activate this function, I will have it give me the total number of branches, the length of the branch, and this says number of branches. This should say number of branches, the number of branches within that dendrite. This is a really neat tool, and something that can be useful when we're looking at this is if you're not sure what exactly you're looking at. Did I graph away? Yep, here it is. You can go in, if you double click on any of these, it will highlight which object that is. So this can be useful if you're trying to find objects within your image that you're not sure exactly what it's referencing. So that's sort of a super basic way of parent measurement. Again, if we double click on this, pretty much all of our measurements, and at this point, I've moved on to the data management section. Most of the stuff is going to live in this measurement tab and data management tab. As you can see, there's a ton of stuff in here. This is probably a good place to start accessing the help menu, especially when we're looking at like accumulating records, aggregating rows. It's just how we're going to organize our data into more easily digestible tables. And we can also do some of our statistical tests down here. We can measure tons of different things from these features. In this case, I had it give me the parent ID, which in this case is the branch, the dendrite itself. I had it give me a line measurement. So that's going to be under parent size and length, line length. One of those will probably work really well for me. You can click these in. You can add as many as you want, and it will update the table for you. I don't know what all of these actually are, so I don't actually want them. We can add, remove. We can hide things if we don't necessarily want to see them. So here I did a parent line length. I did a children count, so I wanted to count the number of children per our parent. And then I did a length, parent line length. So that gave me that information in a nice method. The other thing I kind of wanted to show here was that we can use a filter, like we can filter objects, we can also filter our records. So for this, I had it just get rid of anything where the length of the dendrite is less than 15. I have no idea if this is actually would be something that you guys want to remove, but just to sort of show what this function can do, it'll get rid of any of those like little tiny dendrites that, you know, might not be of interest of us. And then if I'm curious about sort of what those look like, I can order them. So sort records. If you double click, we can sort based on, you know, ascending, descending, length, the branch ID, number of branches, and look at our data that way. So this will organize it from the largest dendrite to the smallest dendrite. And from there, let's say I may be interested in plotting that data. So I can come into my results and graphs and pull in a scatter plot. I can attach that scatter plot to our record. And when I right click on that, it will give me all of the options. What that actually will look like is it will open up a new tab. Let me see if I can zoom in just a little bit. That looks okay. It will open up a new tab in my analysis results where I can plot, for instance, the length of the dendrite versus the number of branches. You can plot.
You should zoom out. It looks
I see that.
Yeah. I don't know what it's rendering right now. There you
Let's
go.
see. Does that look any better? Yeah, that looks a little better.
Flat.
There we go. Sorry about that. So we can plot pretty much anything we want from the data that we have. We can go into our data tab. We can pull in different features, stuff like that. Moving on to the measurement side of our nuclei and Psi3 channel. Let's turn these off. Go into here. Yes, thank you. So let's look at our Psi3 and our DAPI, and we don't want to look at this one. And we'll look at our having and not having. This is an object count that I pulled in. You can see that it's connected to a lot of different stuff. I wanted to be able to measure from the binaries, from my having binary, which is my cells that do contain Psi3, my not having binary, which is cells that don't contain Psi3. I wanted to be able to measure my soma. And I also wanted to, is that all the ones I have? 1, 2, 3, 4. Oh, and I also wanted to measure the total number of nuclei that I had, if that was of interest to me. It'll also ask you to connect channels. What it will do is from the binaries, it will measure the channel within that binary. And usually we like to connect it to the raw data itself, because that's going to be our, our ground truth data. So if we look at this table, we can see in our records, I've gotten a simple count here. I had to count the number of cells that do contain Psi3, the number of cells that don't, and the total number. We could do whatever calculations we want off this. And then I had to count the soma objects. If we wanted to get more information off of the soma objects, we could say, give me the size of our object area. We can have it do object area in the soma binary. And we can have it say, let's say like mean average area of our soma bodies. And that data will populate here. So we can get pretty creative with the sort of data that we want to measure from these. And then from there, in order to sort of organize it, I did what we call an append records. Again, this is a really useful tool for getting multiple tables together. And what that looks like is this. This one will look a little bit funky because it's probably not the best. We have data of different sizes, but it will put all of the data that we got into one table that I can then use. I could put my scatter plot onto this. I can make multiple scatter plots from here. I can export all of this data to Excel or export it as like a CSV file, things like that. And the final thing that we have to think about when we're making a GA3 is what information we actually want to save. That's going to be these little white things at the bottom here where it says save. So for this save pictures, currently it's just going to save the all channels of the picture that we gave it. This is probably fine for me. If I wanted it to save the processed image, I could have it connect to my 2 processed channels as well as my DAPI channel. Next, we're going to decide what binaries we want to save. I don't want to save all of these binaries, and I don't want to save the background threshold. So you'll see I only have connected my having, my not having, my soma thresholds, and my branches and my dendrites. This is a lot of binaries, but it's all the relevant information that I personally would want from this data. And then from there, finally, we're going to select which graphs and tables to save. So here it's currently just saving this table, but I might also want to save the scatter plot. So I can put that scatter plot here. And this is now a fully formed GA3 recipe, or fully formed for what I decided to do. If I were to save this, all you have to do at this point is hit run, and it will spit out all this data for you. Hit run. It'll create our binaries. It will pull up our analysis and our scatter plot. And the other thing you'll notice down here is we got a little arrow on our analysis. It'll say that at 3:11 p.m., which is right now, we ran this analysis on this file. This, as you run more GA3s, this is where all the analysis runs for all your GA3s will live. So this is where this is sort of going to be. And we can talk more about this layout if you guys have questions later. But I think for the purpose of getting through this, this should be pretty good. Does that answer your question? I know we were trying to get to a different meeting, so hopefully we got everything that you wanted to see.
Yes, this is Chi. I think our group is the one that provided you with these images.
Yeah.
Yeah, in particular, I think the RGC counts look great.
Okay, cool.
They're
Excellent.
quite impressive, given how much background there is. I did ask Kale to share with you guys an image that has a decent amount of background to
Okay.
see whether that sort of gets in the way. So it's good to know that that doesn't seem to affect it all that much.
Yeah, sure. Yeah. And again, like we talked about earlier, it's great to give it the best data that we can, but we can't always give it great data. So there are a ton of things that we can do here in order to sort of manipulate the data a little bit and get better sort of nice results out of it. Yeah.
So one of the question I have is, given the manipulation you did to this particular image, can you just apply it to future images, or will that
sure.
alter in the amount of background that's there, for example?
So if this is essentially a pretty well done GA3. I did only test it. I believe I only ran it on this one image. I know you guys gave us a couple. So what I would do, I mean, we can test it. So let's exit out of this. We won't save that for now. Let's see, hopefully this works. Yeah, so generally what I would do for something like this is I would find like a good representative image. I would build my GA3 out, and then I would test a couple other images and just make sure, like if I need to do any fine tuning on some of like the background thresholding, I would do that at that point. Generally, for all of our fluorescence images, we're gonna have pretty similar data, so we shouldn't need to adjust it too much. But I would check it, make any little adjustments I need, and at that point, it should be pretty good to go. It will be dependent, again, if you have some stuff like, I know some of these images, there are some images where like there are some chunks that are a little bit out of focus. Oops.
Yeah, that's true. Because we don't use focal for these. Yeah. Exactly.
I think it's going to struggle a little bit on those. I just don't think it will be able to segment out some of those nuclei so well, but for the rest of the data, for the rest of the image, it should do a pretty good job. And there are ways that we can sort of, without getting too much into it, because it can get a little bit heavy for like an intro level thing, we can make wizards where you can give some user input in the beginning and adjust, like if you can't find a really, really good threshold that works on every single image automatically, we can put that threshold step into a like user defined section in the beginning so that when you run a analysis for the first time, it'll have you do some minor thresholding adjustment, and then you can run the analysis. So it'll still give you the results, it'll still give you the same results, but it'll require a little bit of user input in the beginning.
Got it. Okay, thank you so much for moving this up. I appreciate it. We'll be in touch.
No problem.
With again at the end, just a touch base with everything. Thanks so much.
Yeah, sure. All right. All right, so we will now go from analysis 5 to analysis 1. I'll pull those images up here. This is the one that I want to start with. Okay, so for this image, this one is pretty straightforward. All we're looking to do here is count the nuclei in this sort of central region of our image here. This one's a really good example of segment objects because what we're doing, because we have very little, we don't have, well, not very little. This is not a super high resolution image. You can see at the bottom here, I'll zoom in a little bit. This image is about 640 by 480 pixels. And when we zoom in, one of the first things that I noticed is we have a lot of places here where our nuclei are touching. And this is going to be difficult to segment out traditionally. We also have some like really dark sections and some really dim nuclei of interest. I wasn't quite sure what exactly we did and didn't want to include here. For instance, like really small objects in here, or if these 3 objects are actually one object. So that's something where you guys can probably go back and do some fine tuning yourself. But this is a good image to kind of show off the segment objects feature. So I will open up that analysis here. To check any of your analyses, you can just find the analysis that you want, hit edit, and it'll pop it right up. So if we turn our preview on, you can see the first thing that we get here is our image, and we also have this bar chart that I made. This image is going to be a little bit different because I wanted to kind of show that there are maybe some different ways to get very similar results, different things we can do, as well as how to like do some data management for one of the first things. So this object count bar chart here, we'll get into, I'll mention it, and then we'll sort of get into what it's actually doing right after this. I did 4 different thresholding methods here, and I wanted to measure how each one was doing. So all the way on the right is our manual thresholding. This is the one where I went in and I hand drew each of these little object binaries. And this is the binary that I used to train the AI on, which I'll show you kind of at the end of this section. The AI thresholding is the purple one. This did a really good job. It found a lot of the objects, even in some of those hard to identify spots. The next one I tried was called dark spots. This is one of the segmentation things that we can do. I found it when I was kind of playing around. It's not a function that I had used before, but I thought it might work well for this. And then the last one is what we would have done if we were doing like a standard thresholding. And I'll kind of talk about that one first. So when I was making this image, I'll drop these out for now. When I was looking at this image, the first thing that I noticed when I zoomed in was that they're sort of a little bit fuzzy around the edges, these nuclei. So I wanted to see if I could do something about that. And pre-processing, you can see I have this pre-processing section here. This is one of the ones where I kind of play around the most with and see like what I can get, how I can make the image a little bit easier to work with. Some really common ones are contrasting and sharpening. So that's what I did here. I ran a contrast on this, and that's already looking better to me. We can see that our whites are a little bit whiter and our blues are a little bit more dark blue, and that's going to make the segmentation easier to begin with. It still looks a little bit blurry to me, so at first I tried to do just a regular sharpen, and you can see that starts to look really over processed and kind of not exactly what I'm looking for. So I ended up taking that out, and I used instead a sharpen slightly. This looks way better to me. It's looking maybe a little bit processed, but I think this will be way easier to threshold out. And that's kind of what I found when I was doing this threshold. And it's nice, kind of like we mentioned before, sometimes we can work backwards. The first thing that I actually tried was without any pre-processing. I just attached this threshold directly to my RGB channel. And I was just really struggling to segment out these nuclei from each other. So I decided to put in some pre-processing, and that ended up helping a lot. You can see we have our bar chart here. Oops, I dropped you out by accident. You can see that when we have our nuclei here, if we right click on any of these, we can disable them and see kind of how they're affecting our image. And you'll notice that we can get live updates on results here if I don't do that. So if I disable this, you can see I get a significant drop in the number of cells that I'm picking up for my standard thresholding. So that tells me that I probably want to leave this in. This is going to be a lot more accurate, and it's going to give me better results. So that was my pre-processing. I then ran a standard color threshold on this. We'll make it a little bit easier to see. Those settings are all here. For thresholding, generally it involves just some playing around with your settings to try and get the best threshold you can from a starting basis. And from there, you can see I have a lot of the things that we've been talking about. I have an open function that I used. I tried to dilate them a little bit. I tried an erosion here because I thought that would help sort of segment them out from each other, but I ended up just getting really awful counts. So I kept the dilation in here and I ran a separate objects. And that does a pretty good job, actually. It's not terrible. It segments out some of these that were connected initially, but we can still see it's missing some of these like lighter objects, and it's still connecting some things that we probably don't actually want it to connect. The other thing that you'll notice is when I do this threshold, I'm getting a lot of this like stuff out on the periphery that we end up really not actually wanting. So I pulled in an ROI that I had made earlier, and I used the AND function. If you'll remember, the AND function is going to return the binary of only where the two binaries that I connect it to intersect. So when I activate that, it will only give me objects within this region of interest that I'm concerned with. That helped a lot. I didn't have to go in and manually remove all of these objects from the periphery. And going back to that color by ID, I pulled one of those in just to see if I could get a better idea of what it's doing. And it doesn't look terrible, but I think that we could do a little bit better. So the next one that I went with is our dark spots detection. That is a function that lives in spot detections under segmentation. What dark spots is going to do is, let me pull this up real quick. What dark spots is going to do is it's going to try and find any dark spot of some diameter, and we can tune those parameters here to try and get it a little bit better idea. So if we see what that's doing, it's pulling a lot of points from all over my image. But when I went and looked at the region of interest, I found that I thought that it was doing a really good job of picking up some of those spots that the standard thresholding missed. So I used my AND function again to try and get rid of some of those objects. And this is looking pretty good, but you can see down at the periphery here, I sort of have some of these half objects that got cut off, and I wanted to try to get rid of those. So I ran a filter objects to try and see if I could work on that a little bit. And so for this, I did a fill area of anything less than 20 got removed. And I did like a mean intensity. So to try and remove some of these like really, really light ones that actually wasn't of interest to me. And that does a pretty good job. We're still grabbing like this, maybe we might be interested in, I'm not sure. But like, I don't think that some of these really, really light regions are maybe still interested in. So this one I thought did pretty good, but I wanted to sort of plot it out and see if the data actually looked at that way. But what I found is that this one tends to overestimate the number of nuclei that are in these cells. So it ended up not being the best solution. The really nice one is this one that we did with our AI thresholding. You'll notice that aside from, let me see, can I zoom in a bit on that? Does that look good? Yes. Aside from this color by ID function, which again is just going to relabel them, this is the only function in this method of thresholding. And when we look at our whole image, you can see that it does a really good job of picking out only the nuclei in this central area here. I didn't need to use the AND function to get rid of some extra stuff on the outside. I trained the AI from my manual thresholding here. And if we go and zoom in, this might be a little bit difficult to see, but you can kind of see the pink is going to be my segment object AI, and the red is going to be my manual thresholding. So it does a really good job of matching up. And even in places like here, where I missed some of these edges, I forgot to, I missed removing these sort of border objects. It doesn't include those in the AI thresholding. So it is relatively intelligent in how it's segmenting out these objects. So I was really happy with this one. This is actually the second AI that I trained. The first one that I trained was it grabbed like 3 or 4 of these little objects on the border. So what I did was I removed those and I retrained the AI, and I'll show you guys how to do that as well. But this was super powerful. I was really happy with how this came out. I did the color by ID just to double check, but it really didn't capture maybe 1 or 2 that are like really squished together, like multi-nuclei. So this one was really good. I really liked how it worked. And then obviously at the end, we have the manual binary that I trained it on. From there, in order to get this graph, I went down here. I did an object measurement. I did the same for all 4 of these. So if we look at that, I did an object measurement. I connected it to my channel, my RGB channel, and my binary that I made. And when I look at what that gives me, you'll see that it's going to label every object ID. This is how I had this set up. I just had it give me an object count. You'll notice that this loop indexes is in all of the object measurement features. That's for if we're doing things like time loops, which we're not doing here. So I tend to just hide it. for most of the graphs that I use, but it can be really useful when you're starting to work with larger data sets over multiple time points. I just have the object ID and the total object count here. As you can see, this is going to give us every single object as well as the count of all the objects for every object. This is a lot of redundant data that I really don't want to deal with. So I'm going to do something that I use a lot called aggregate rows. And what that's going to give me is one count with all the objects of this image. If we look at the options for this, you can select any of your columns, you can select which aggregation you want from it, and then you can have that plot to a separate table. And once I got all 4 of those values, I put it into an append column to give me this nice single table with all 4 of my values. And from there, I did the same thing I did with the scatter plot from the previous one. I had to give me a bar chart, and I made them some nice colors to represent the binaries and things like that. Any questions about sort of this particular analysis, how I did stuff, how things are organized, that sort of thing?
Yeah, this image came from my lab, and it looks like the counting went really well. You counting photoreceptor nuclei in a toluidine blue stained plastic section from Brandon Anderson. I'm just wondering how you did the initial marking for to train the AI.
Yes, great question. I am about to do that next thing. So let's get into that. There's a couple of different ways that we can do thresholding in elements. We'll save that. We don't need to save that particularly. I will close this for now. Oops, wrong image. That's our training data. So this is the image, the raw image that we got. I'm going to go over these binaries. I just put them in there so I don't have to actually draw them all. You'll see in elements, there's a couple of ways that we can make binaries. I like to pull in the binary toolbar here. This is going to give me a couple different options. So for this one, I really didn't want to have to go in and like actually manually draw all these little objects because that would take forever. Whoops, I put that on top of one of my other binaries. Do that. So what I ended up doing was I pulled in my thresholding. And this is the thresholding I use just as like a base layer. And what I can do is once I get like a threshold that kind of looks decent to me, I can pull it in as a stored layer down here. And you can see, again, we're running that same issue where we get a lot of this background stuff. So I drew a second binary, which is just my object of interest. And I came in and I did a binary operations. That's going to open up this dialog up here. And what we can do is I want to take the threshold that I used. I want to take my ROI, and I'm going to do the AND function. And that's going to give me this nice region. This is a really good starting point. I've done that already. From here, what I would probably do, or what I did, is I would go in with my binary toolbar. I'm going to zoom all the way in. And I'm going to start working on segmenting out each of these objects. You can see here, we have like 3 nuclei. I'll try to make this a little bit easier to see. We have these 3 nuclei here. We have a really nice tool called Separate Objects Manually. This is going to be a bit of a tedious process. It took me 30 minutes or so to get like a pretty good binary. You can hold and manually separate all of these objects that it thinks it's getting. So this is what I would do. We can remove objects I think that maybe we're not interested in. We can draw objects on top of it. So I'll left click, left click, left click, left click, right click to make an object. And that's how I would go for this. I would manually go in and make like fine tune this to get it really, really nice. This is going to be the most time consuming part when you're going to make something like segment objects. I was talking with our software team, who's the people that sort of designed these, and for segment objects, they recommend giving it about 1,000 objects that you want to train it on. This image had about 450 or so. So I would train it on 2 of these images. And that's going to give you a really good starting point. You can see we did a really good job with just 1 image. So you're not going to necessarily get bad results, but in order to like fully optimize it, 1,000 objects is probably what you want to shoot for.
Great. And if you want to modify just a little bit, like you get your final count, and and the AI it's not counting some nuclei that are a little bit dim, and and maybe you want to just tweak it so that to teach it to count those. How would you do that?
Absolutely. So we can come up here. Let's run one of these. So if we go to our NIS AI tab, can everybody see that? Yeah, okay. This is where we're going to do all of our training and also all of our segment. So I can come up to here to segment objects. I can point it to from a file that I select, which I know lives here. This is the one I trained, OS40X. I can train it. I can run it. It will make a new binary layer for me. So I'm going to store that layer. I had it give me nuclei round 1. It always does this weird lookup table thing. So this is the binary that the AI gave me. If I want to zoom in on this and modify it, like, ooh, here, perfect example, it accidentally grabbed a double binary. I can manually edit this, and I can save this binary. So I can go in and retouch up anything. Sorry, I'm probably looking at the wrong camera. I can go in and manually touch up anything. And this is also how I would retrain the binary. So if you find that it doesn't get it quite right, I would, go in and let's say this is 2 cells, maybe. I would go in, touch up this binary, and what you can do is go to NIS AI, train segment objects. You'll get this no matter whether you're training it for the first time or training it again. What I can do is take my RGB channel. I can take let's save this image first so that it stores these layers. Saving. Oops. All right. It did not like that. I think I was playing with that file. It may have been corrupted at one point. So I'll open that back up. But you can go in and you can retrain the binary and then retrain it on that AI that you had initially trained to sort of fine tune it a little bit if it doesn't get it quite right the first time. And then even then, you can always go in and manually change those binaries yourself to whatever you want them to be.
Great. Thanks very much.
Sure. Okay, any questions on that analysis specifically before I hand it over to Lauren and she talks about some of those other ones? Excellent. All right, I will hand it over to Lauren. I will close out of this.
All right. I'm going to take over. This we're running a little long, and I unfortunately have a hard stop at 4. So I'm going to go over very specifically. We had a group that was looking to do a convert. So I'm going to go over how you do that and what that kind of output is going to look like. And then I'm going to go into one of the other thresholds that does a little bit of parent-child comparisons. We're going to be looking at that one in 3D, but I also did a max intensity version of that as well. So if everybody can see my screen, this is an image where we have a transmitted light picture. It may be that you want to use transmitted images to create another image that contains fluorescence in it. This one specifically, we were looking to get nuclei, so image of nuclei, but based on the transmitted light image itself. So, a human can look at this and say, I know where the nucleus is in this. But an AI needs to be trained in order to get that information out because grayscale, everything's pretty much the same intensity, and intensity-based thresholding on a transmitted image is inherently difficult to do. So for converts, what you want to have is an image of what you would like to produce from the convert and have an image of the channel that should be producing that final image. So in this case, we have both our DAPI and our transmitted. We want to give our train convert this file. We're going to want to convert from transmitted, so the transmitted channel, to produce an Alexaflor channel or the nuclei channel. This would be one where you would continue training, run it, and then hopefully produce a result. We gave this just a center transmitted picture. It's going to do better with nice, high quality DIC images. Out of focus DIC is probably not going to produce the best picture. So we've already created a convert that lives over here. I'm going to hit play on this. It's going to point to the transmitted channel, and I'm going to let it process. These can, again, like Nick said, be put into your GA3 routines. So you could have it create this DAPI channel to threshold off of and use that in your final sample. So you'll now notice that I have 3 channels in this image after running this GA3. We have our original DAPI channel that was an acquired image. And then we also have this Alexaflor AI. This is the artificial image that has been generated off of the transmitted pictures to create this DAPI image. I'm going to change the colors on these. This one green, and I'll make this one red. So we can see how well it did when we overlay these two. When you're looking at this, you can see how accurate it is in trying to generate that DAPI image from just the transmitted file that we gave it. One thing to keep in mind is I gave this, a center image and I didn't train it on a ton. So we have some areas where you're up near the top of the stack where you have, images that weren't, it's created the green from something that it thinks it saw in the transmitted channel. So if you truly wanted this, you would have to run it on every single layer in the stack. And it also doesn't do that great down where we have maybe more muddled DIC images and it's created these kind of odd structures. This would be one where I would then give it an image because there's not a lot of red in this channel. So it's probably picking up some random things in here, and we would use this as an input to then continue training this algorithm to try and clean up things like this, where it's come up with stuff that it thinks happened, but it was trained on a nice, crisp, clean center picture. This is one of the easiest algorithms to train because you take a picture and then you compare it to another picture. Enhance is going to work exactly the same way. You're going to have an image with 2 saved channels in it. One, which is the low quality image that you want enhanced to a very nice quality image. Train both of those and it will run exactly the same. It will create the enhanced picture out of the lower quality image. So these ones are much more easy to train than the segment where you have to be very specific in the binary that you are generating. Does anybody have any questions about this convert or enhance? They are the easiest ones to play with. You basically just need to give it pictures in order to train these. Okay, I'm going to move on to another set of images. And this is where we're going to play with some more stuff. So this was a data set that Anu gave us. It's a 4-color. We have very nice delineated DAPI signal. We have a FTSE channel that produces these cute little bubbles inside of the image. Get this so we can see it a little better. Yeah. There's a TRITSI channel that is also creating these liposomal structures. And then there's this Sci5 channel that has a kind of interesting pattern where we get a lot of labeling down near the bottom of our Z stack and then it kind of disappears on us up near the top. I'm unsure if this was a cytosol stain or something like that, but it is kind of a diffuse structure, and it doesn't necessarily overlap with a lot of these red and green structures that are higher up in the cell. I believe the idea of this was to get some sort of count on green and red for each cell, maybe using this purple channel as the outline of the cell, or maybe just using nuclei as a comparison. There's a lot of options with this. One of the routines that I did make was trying to use a maximum intensity projection in order to create a, do an analysis on this so that we could get purple sort of in the same layer as the red and green channels. This is a great way to do the analysis if you don't have any of the 3D features. You guys currently don't. I know we've talked to Anu about expanding those, and I am going to show you what it looks like in this one because it is very cool. But I made a maximum intensity projection GA3. The nice thing, yes, the nice thing about GA3 is that you can do some of these max intensity processing on the image in the GA3 routine. So if I wanted to save out a maximum intensity picture of this, I could save out all of these images that we've produced from a max intensity. Maximum intensity, just like Nick said, when you click on this little button, you can have it do all of your Z stacks. If you had time, it could do it in time. It's a weird view. But if you right click, it will show you where this is located in the help file and what it means. And if you're trying to find it, it sits in ND processing specifically. So this is modifications that you're doing to the ND image itself. This includes things like maximum intensity if you want to average out a bunch of frames, if you want to change the bit depth, you want to render an RGB into different colors. All of those live in what we call ND processing that are going to affect the ND image itself. Once you've done this maximum intensity projection, you can then threshold on the maximum intensity. And this preview is taking a while. When we move into the volume, I'm going to show you this length of time that it takes to reduce this is why when I'm doing a lot of this initial analysis, I tend to crop files down so that you have a smaller data set to actively see the preview. I made a whole bunch of binaries in here for the different colors. These were all very rough, just based on general intensities. I didn't do any pre-processing, didn't do any cleanup, but we can get a lot of binary IDs where we are looking for, if I can get it to show up in this image, how many FTSE channel we have related to, and I did this to the nuclei. For this type of build, you're just essentially copying things over, individually thresholding, and then getting a saved table out from this analysis. Most of these thresholds were just very simply done on the intensity. I didn't do any pre-processing or anything like that. So you can have routines that are incredibly simple, where you just hit a button, it does pre-processing, so you don't have to create a new image. You don't have to do this every time. You want it to create a max intensity, you want it to run a decon, and then you want it to do a very simple threshold. People build these types of GA3 routines all the time so that you hit one button and then it is done. What's fun about GA3 when you have our volume analysis is I have a cropped version of this picture. So I have cropped it just in XY so that I have a smaller number of pixels that the 3D volume has to render in order to create the preview image. Otherwise, you're going to be sitting here waiting for it to continue to re-render your picture. So you can see from this volume that the purple it's really bright near the bottom and then kind of becomes this hazy structure up near the top. When we're looking at it straight down, There is maybe some information in here that we would be able to glean out, but the easiest channel to threshold in this image will be the DAPI, as you have these really nice bright signal locations and then black backgrounds. So if we go into this analysis where it's looking at the volume, this one is a little more complicated. I'm going to go section by section. For DAPI, I don't think this needs any pre-processing. You may decide that you do want to do some of that, but I just decided to do a 3-dimensional threshold. You'll notice that when it is able to do 3D, you get a little 3D signal right here. If you want to find what the 3D features are, once you have it, there is an easy turn on button up here that will only show you functions that work in 3 dimensions. It's important to remember that this is working off of your graphics card to try and render these in 3 dimensions, and so sometimes the previews will take a little bit longer. So if we just look at our DAPI channel, and we want to look at the thresholding for DAPI. For some reason, it made it blue, which is pretty difficult to see. Usually it tries to choose a contrast color so that you can see your binary on top of it. One thing to keep in mind is you may hate the color that it chose. You can always right-click on the threshold channel and change this to a nice contrast color. I'm going to make it orange so that we can really see where our threshold shows up for the DAPI. Color by object also works in 3 dimensions, and just like Nick said, it's really helpful to decide, is this creating one giant object, or are there multiple objects in whatever it is that I'm looking at? When you hit color, it should make it a fun party pattern in 3 dimensions. You do not have to save out the colorized binary. I'm using the non-colorized one to do a lot of the analysis, but it is a nice way to just double check if the binary is doing exactly what it is that you would like it to do. I have these binaries rendering in this kind of a grid pattern. You can change a lot of that over in this section. There's not the equivalent of opacity in volume. It either displays it as this kind of outside grid, or you get a solid voxel-based structure that is opaque. For this rendering, I chose this just so that we could all see where the actual channel was and compare it to the signal underneath. Now, if we go back to the Sci5 channel, I'm going to turn off the DAPI. And you'll notice that when I changed the channel here, it selected the same color in the GA3. So it is trying to at least be helpful and show you what it is that you just selected. When we look at this, from one side, you can kind of see we have delineations between these cells, right? I can tell that this, I can see, can everybody see my screen?
Yes, I can see your screen. Maybe just try rejoining McHugh.
Okay. You can see where there is a extracellular label that it seems like I have cell structures in here, what I would assume this is labeling as a cell. When we look at this from the top, however, it kind of looks like a lawn of fluorescent information. In pre-processing, one of my personal favorite is a rolling ball correction. What this is going to do is it's going to take every single pixel, it's going to average an area around it of intensity and then subtract that from that center pixel. You can decide how many pixels wide that subtraction is. And so this is essentially like if you mowed the grass, anything that is within the top layer of grass will disappear, but your structures that you really care about, like, I don't know, your garden gnome, will start standing out better against the grass level in the background. When we do this rolling ball on our image, you'll notice we get much more clear outside boundaries of where this cellular label was for the Sci5 channel. This may not be as pretty as you want it to be. So this is where multiple pre-processing becomes kind of critical. I did a simple smooth to see if I could kind of even out some of this kind of bumpy background. If you're just using this to get a general idea of where the outlines of your cells are, this is a great way to just remove a lot of muddy out of non-specific signal that you don't want to include in a binary that you produce from this processing. If we look at what this does specifically to the binary that is produced, we have a Psi3 threshold. It's specifically looking at this smooth channel. This threshold seems to have produced a pretty nice render on where all of this cleaned up green, purple label is. We can kind of see we have nice clear boundaries in the center here. And some of the structures have some interesting kind of internal structures where I assume they're in, it didn't label fully throughout the entire cell. If we compare this to another threshold that is done directly on the unprocessed color, we get something that looks like this. This was doing exactly the same intensity-based thresholding, but on a pre-processed image and a non-processed pre-processed image. So making decisions when you're doing this analysis, I'm going to try and crop in a direction. You can kind of see how you get a much more amorphous blob, and we get a lot less specific labeling when we do not do any sort of pre-processing on the data that we're giving the threshold. For me, I would think you might want the green label. That may not be what you are interested in. You may want to know everywhere that purple exists in the cell. So making these kind of decisions is going to be a little iterative when you're first starting out building these routines. The software will do exactly what you told it to do. And so making a decision on what it is that you want to see out of your data is going to be very important. The two other channels that we had in this image were very obviously, coordinated with each other and labeling a very similar structure, at least from how I was looking at this. So if we stop looking at our Psi5 channel and add back in Tritzy and Fitzy, and get rid of the Fitzy channel does have what I would consider some non-specific labeling. If I was going to compare it to only where Tritzy was. I, again, decided just because it cleaned up and made the thresholding easier to create a rolling ball on this image. You'll notice that a lot of that kind of non-specific background label, which could very well be labeled, it's just a decision that I made for this data, disappears on us. This is much easier to threshold for these kind of globular structures than I would consider this original channel to be, to get all of that information out. I also decided that, if some of these small spots near the bottom were actually pretty important, the rolling ball may have reduced their prevalence in the image. So I decided to add just a local contrast to this and see if maybe that brought out any other information. When you're looking at your own data, you may decide, I do not like what local contrast did to this image. This is why it's really easy to be able to switch back and forth between these types of things. If we hate what local contrast is doing, you can easily right-click and delete it, and then only do your thresholding on the rolling ball. If you have something linked in a chain and you delete it, it will automatically link whatever was linked to the bottom of your most recent, whatever you were deleting, to the one above it. So you don't have to redraw all of your lines every single time if you're going to pull something out of organizational structure. For FITSE, firstly, I made a threshold on FITSE specifically. Green really quick so we can just see that. I was using the really bright center spots because when I started to drag this out a bit further, it got a lot, you know, much more structures showed up in this process. So I just did a simple dilate. Again, this is going to be up to you on if this is the exact structure that I want for this final channel. You can change the number of times things are dilating. I did it 4 times because I thought it looked nice, but you could do it 3. Most of what I was trying to do on threshold here was so that you guys could see what it's doing in 3 dimensions on the screen, which I know is a little bit difficult when you're not right in front of the data that you're looking at. The final channel that we played with in here was TRITSE. For the TRITSE threshold, I think that this has some pretty nice distinct bright versus dark areas. There is maybe a little bit of background, but a traditional threshold should be able to handle this pretty easily. I ran a threshold and then also did some processing on that image just to try and get, again, a kind of easily viewable version of the binary over the red channel. One thing that you can do that we have not mentioned yet is that at any point in doing any of these modifications to, you know, adding a dilate, doing something like this, you can create a new layer in the binary list of that processed version. So what I've done here, and let me see if I can zoom in. Up until this dilate, it is the same binary. So it is named exactly the same thing. It is the same color. Everything that we're doing to it is being saved on that same binary layer. I decided, you know, this kind of looks like some of these have created one giant blob in here. Try and zoom in so you can see that. Maybe we want to separate objects out from this, but I don't want to fully modify my original binary. I want to keep this dilate. You can right click on any of these. And then it's really hard to see and labeled, but right next to the little A where you can rename it, if you wanted to call it something specific, you can make a new layer. And what that will do, it will create an entirely new binary layer that is separate from, but based on the processing that you've already done. So this separate objects, I made a new binary layer of it. I made it yellow so that it would show up compared to our orange. You can then do processing on that binary. So you can create quite a lot of binaries within the same image that you're doing analysis on. And you can go in and select a bunch and delete them if you hate how it did processing on those props. So the final binary that I ended up with for this TRITSE channel looks something like this, where we have some very small internal structures that are labeling, but we're trying to kind of eliminate some of these other out of focus things. That may not be what you want out of your final data, but it was something that I thought would look pretty nice when we start doing representations of the binary operations, which is what I'm going to talk about next. I'm going to switch over to the DAPI channel because you can see all of these fun little guys much easier. When we look at our pink and yellow binaries, we may want to know where these intersect, which of the pink binaries have yellow sections in them, which of the yellow have pink sections in them. I want to know all of the binaries combined. That is where, as we talked about previously, these binary operations come into play. I did just a couple of them. I'm going to turn off pink and yellow just so that we can kind of see these other colors a little easier. And questions. So if we wanted to know where pink and yellow both exist in space at the same time, we would do an and. That would be this kind of light blue binary. So wherever this exists in 3D space, we have both pink and yellow also, they exist together. This should be a comparison to or. Or is when you have both are existing and we are combining them both into one mighty binary. This is a nice way to, if let's say you have, you didn't want to go through the whole process of segment AI, you had something that was really bright and really dim, you wanted to do 2 different thresholds and then combine them into one. This would be the way that I would do it. You would do an OR and create one large binary that can have a variety of different threshold intensities in it. Having is one of those ones that Nick very specifically said is directional. So I have 2 different versions here. This one is whatever this first dot, it's this binary. So my yellow binary having pink. So wherever we're going to get any of the objects in yellow that have part of pink in them. And that's going to be this representation here. This will be contrast with the other way that we have this, where the first part of this binary is being directly pointed at the pink binary. So any structure in pink that contains yellow would be this render of purple. And you can see they are different sizes and different shapes. This should match up with items that we have in pink in volume. And you'll see that we have some inside of this structure render that just didn't have any of our yellow binary in them. And so they do not show up in our final binary for having. Using these binary operations can be cumulative, so you can do them multiple times on multiple binaries. And they're kind of, they're a great way to get an output that might not necessarily be easily done with a single threshold. So, turn that on. Nick briefly mentioned this earlier, but I kind of made the assumption that you might in this analysis, threshold, there we go. You might want to know, how many pink and red objects there are close to a nucleus. So we call those a parent-child relationship. These live in measurement. There's a children one, so this is if you want specific information of the children. And then there's a parent one where you want specific information related to the parent. So how many children per parent? Children gives you slightly different outputs for, I want variety of different information about each of the children for a single parent. I just did parent measurements on these. Click open, there we go. There are a lot of different sizes, shapes, number, position, relative position. A common one is this children count. So how many of a certain thing do I have that are near my set parent? In this case, parent is the nucleus, and then I made 2 different ones where the pink channel was one and then the yellow channel was another. You can rename these over here. It will just throw in a random name on this. So it will just call it something where dappy fitsy based on the binaries that you've given it. You can easily rename all of these. Another thing to keep in mind when you're doing parent-child relationships, and which is one of the more important that you would want to keep in mind, there's 3 different ways to think about children. One is the children are entirely contained within the parent. So this would be if you're doing nuclei labeling or something like that, where you're doing different spots inside of the nucleus specifically. So you want to know that the child is entirely contained within the parent. There's child intersecting parent, so this will do both any child that is inside the parent, and it will do any child that is touching the border, like right next to a nuclei. This could be, you know, lysosomal tracking, things like that. And then the one that I chose for this, which is child's nearest parent. So whatever is close to it. It will also include things that are inside of the parent and are touching the parent. When we get these type of records, we end up with something like this, where we'll have information per nucleus. One of the things that we have found is with two cells next to each other, in the help file, it describes how it's deciding what the nearest neighbor is, and I don't know that math off the top of my head. But that is a great point. It should be easily accessible when you click on the help file, and there are some pretty nice walkthroughs in the help file of parent-child relationships, because this is something that I think a lot of people are interested in. So per nucleus, you get FITSI objects per nucleus and the volume of them, and also TRITSI objects per nucleus, because I did two different measurements, one for DAPI as a parent and FITSI as our child. As Nick mentioned before, but I would like to reiterate, we strongly recommend doing all of your intensity analysis on a non-processed data. So you can, Elements will let you do analysis on something where you've done a rolling ball and smoothed in intensity. We don't really recommend you doing that. It's not going to give you an error. It's going to say, okay, I'll do exactly what you want me to do. Keep that in mind when you're building these, because you could link it to something where you've done some sort of process to it and not be getting accurate data out of it. And then you can append the columns. So when I run this, I should just get a table out. That's the only thing I've segmented this towards. I don't need any of the internal binaries. I'm not interested in them. And then I wasn't going to do any modification. Like, I don't need the modified images out. It's up to you how much information that you want to save. You could save every single one of these binaries into the final processed image. You could save out the processed images, like if you wanted to save out a max intensity projection. It's all available to you, but you have to remember that you have to link something to this bottom section where the save pictures, save binaries, all of that is, in order to get that out finally. Again, you guys don't technically have this 3-dimensional render yet. I know that was a hope and plan for the future, because it is great for these confocal pictures to kind of deal with this stuff. When you do a max intensity projection, you can think that a lot of this volume information, where there might actually be two structures in this pink, might get smushed together. It's one of those things that you want to think about for just long-term processing. Are there any questions about this before I have to run, and I will let Nick take over again?
Hi,
Yeah.
Anubila, thanks so much. This is really helpful. So in the apps right now, since we don't have the volume version of it, does it make sense to just do a single good layer as opposed to having a
You could do, so I ran this
maximum
on I ran it on a maximum intensity. You could also run it on a single layer. That is complete, like, I would test it and see if you get dramatically different pieces of information. When I first started playing with this, I thought I would use this purple value as the like parent, and then decided that the nucleus actually worked pretty well for what things were doing. I would say, because you lose purple really quickly in this, like it drops off where all of your red are. I didn't think just a single layer would be useful for that kind of parent-child relationship, but your DAPI are right on the same level as a lot of your green and red. So it'll really be up to you whether you want to smush everything together and then do an analysis on that, or just pick a certain layer and do an analysis on that. If you run it on multiple ones, you know, multiple stacks, you do run the risk of getting double counts. So that is something to keep in mind when you're doing that kind of analysis.
Thank you.
Yeah.
All right.
All right,
I
guys.
will Pop back over. We'll share this window.
Stop sharing. There we go. All right. Thanks, guys. Have a great weekend.
Thanks, Lauren. Okay, can we all see that? Yes, we can. All right, so this is the final one that we did. I'm gonna hide this for now. What we wanted to do here was, I know, and Anu, when we had initially talked to you, we had talked about measuring sort of the distance between cells to get an idea of how they're interacting with some like drug treatment, I believe, and seeing how, because I think you described they sort of like spread out. When we were thinking about this analysis, we can measure the distance between two objects, but because we have this big field of cells here, one of the ideas that we had, which I thought was going to be a good measurement, again, up to you, this is what we decided, is I think what we ended up deciding to do is what we measured was the area where there are no cells. As we sort of move through this stack, you can see that as our cells get further apart, we get more of this area measurement. And as they get closer together, we get less of an area measurement. So that's what we did here. We tried to keep this one. Not all your GA3s have to be super complicated. Oh, did I turn my camera off? No. Oh, there I am. Okay. So not all the GA3s have to be extremely complicated. If you'll notice, if you look at all my connections here, I ran all of this off the DAPI channel. So I pretty much worked here. The first thing I did with this was I wanted to threshold out all my DAPI cells. And DAPI is usually a really good channel to threshold out from, because if we adjust our lookup table all the way here, we can see that DAPI sort of just kind of bleeds through into everything. From there, I wanted to clean this up a little bit in order to get a better idea. Let's see, I wanted to clean this up a little bit just to get rid of some of these stuff. So I did a fill holes to kind of make sure that I'm filling in all the cells. I did another one of those filter objects that we talked about to get rid of some spots where we might not want cells, and maybe I want to adjust these a little bit. And then I did an erosion just to make sure that I'm getting all the layers that I want in. From here, let's look at all channels. I did something called grow regions. This is also going to live in the binary processing tab. A lot of common ones that we use are like zones of influence, water shedding, grow to intensity in this case. And this is really good for identifying entire cells. So I ran one of those, and you can see that that does a pretty good job of actually really identifying my cells where they are and not where they, and where they're, and identifying not cells where there aren't any. I ran another fill holes here, just in case. I think there were a few places, like if you look up in the top left section here, there's a little hole that I wanted to fill. So I ran one of those, and then from there, I did one of a binary operations called invert, and that just takes our binary and it inverts it. So that gave us a really good binary for just telling us everywhere that our cells, where there aren't cells. And as they start to spread out in this Z stack, you can see that it does a pretty good job. From there, I just moved into my basic measurements. So I ran an object measurement. And from here, I just wanted it to give me, I called it not cells, fill area, as well as an area fraction. So for each, I had it aggregate rows. Just want to live back here. So what this is going to ultimately spit out at me is, because I'm not sure which exact feature we wanted, it's just going to give us the total area of where we don't have cells, as well as an area fraction. And you can see as we move through this image, it will live update. I'll zoom in a little bit so we can see that. As we move through this image, it will live update the results for us. And we could export this, run it on multiple images. And I think that this would be a really good way of measuring sort of this response to a drug of these cells spreading out. And again, I ran all of this off the DAPI channel. So I didn't technically need all the other channels, although I'm sure that there are plenty of other ways that you could do your thresholding to sort of segment out these cells. I know that when I first started doing this, Lauren may have I think Lauren may have adjusted these slightly, but I know she had to leave. So I did try doing tons of like other water sheddings from the green channel and the red channel. And at the end of the day, I kind of just found out it was best to run it all off of DAPI. So I think that this gave us, again, pretty good results. And this is definitely going to be a good starting point for that sort of analysis. And then from this, I just saved the original image. I saved the binary. I probably want to save this binary, actually, the inverted binary. And then I saved the table with all the measurements in it. So this one was relatively straightforward. We had thought about a lot of different ways of trying to measure all the individual lines between cells. And it ended up, this was kind of the best way to do it. So I don't know if that sort of makes sense. And if anybody has any questions about this analysis, and if not, I know that we're sort of kind of running a couple minutes late here, but if anybody has any like lingering questions about GA3, about a specific analysis, now is a good time to ask. I can hang out for a couple minutes and sort of go over stuff. If you want to see more things in software, we can do that as well. How does it deal with two? Many images we'd be analyzing are from knockout models. These models develop various pathologies. So answering this question from Catherine, can the AI detect the absence of something that has been trained to look for? Or do you need to train it on images with pathologies? Yeah, it won't be able to say, you can't train it on the absence of something. For this tool, for our AI tool specifically, you would probably need to train, it's going to be dependent on your samples. I think there may be some ways to do this with standard methods. I know like before we had the cells that have the having and the not having cells. That might be something that we do here without kind of sitting down and looking at your specific images. It's going to be hard to say what the best path would be. But to answer your specific question, we can't train the AI to detect something that isn't there. Usually we're going to have to detect something with some identifiable features. And to answer Eric's question, I don't believe we've benchmarked the segmentation against cell pose. I'm not familiar with cell pose, but for us, each of our segmentation and segment object routines is going to be different from one another. It's going to be dependent on how you train them. I don't know how cell pose does it. But it would be difficult to benchmark something where each AI is going to be the same net, but all the weights are going to be different. So I don't know how we would benchmark all those individual segmentations against another AI-based neural net, if that makes sense. Okay, perfect. All right. Okay, so if nobody else has any questions, we can probably end it there. If you guys start messing around, we have another question. In the case of we need distance measurement between cells, we may want to use the green channel as it's the marker cell lines. Yes, there is a way to do distance measurements. So there's a nice little function we can do. If you search something that you're looking for, usually you can find it. I know there's a function called nearest object that lives in the measurement tab. What nearest object is going to do is measure the distance to the nearest object. Let me see, what does pairwise distance do? Okay, so pairwise distance may be what we want to do actually here, because we can choose, if we go into the function here, I haven't played with this too much, so I actually don't know exactly what it's going to do. But if we double click here, we can see the reference A center to B center. We can also do A border to B border. So that may be, if we can define our cell border really well, that may work nicely for us. I would probably, if I were you, I might maybe say threshold on the green channel. Let's deactivate this so it doesn't pop up every time. So let's look at that channel real quick. We'll turn this off. I haven't done any pre-processing on this, so I don't know exactly what it's going to look like, but maybe we do something like that. We run some smooths to get it just to look nice. And then I'm not sure exactly what this is going to look for. Layer 1, layer 2. So this is going to give us a distance measurement. Yeah, I'm not sure. It's going to be hard. I think the reason that we decided on the way that we wanted to do is you need to, because when we segment these out, what's actually happening is that it's creating these like really large objects. We're not actually able to, we'd have to segment out each individual cell. There are ways to do that, but it's probably going to be a little bit time intensive and outside of the scope of what I can do in like 15 minutes here.
Yeah, that's fine. Yeah, we can work it out and talk
Yeah,
about it
so there
later.
are ways to get distance measurement, because I know when we had talked about it initially, because the distance between like these two cells, they're very close here, but very far apart here. It's gonna be, I don't know of any way to do like 10 or 5 or 100 measurements in between these two cells and these two cells and these two cells. But this will give you the distance from border to border between two objects. So if we can find a way to reliably segment out each cell as its own object, which we should be able to do, there are plenty of ways to do that, then we can use this distance measurement to try and measure the distance between the cells.
Yeah, I was thinking of some way of doing kind of automatic line profile where we look at the intensity signals and there's a break-in signal between the two cells.
Yeah, the way
Something
that
on those lines.
Yeah, something that I would think about for that, maybe, again, if we can, I think the first step for what I would think is maybe if we can do like a nuclei segmentate like threshold. Don't want this record right now. So if we were to threshold these out with a little bit more like that, and get like our nice actual nuclei, we could do something where we find like the center. Detect centroids would work here, where we find the binary and our channel. And if we zoom in here, you can see that that's going to identify a single pixel at the center of each object. And so if we did a distance measurement on that, maybe we'll do center to center, let's say. Let's see where it's going to pull those measurements from. Yeah, so it's going to like, it flashed here. This may be like a good, like a kind of how I would start approaching it is center to center after I can identify like the center of my cell, or after I can identify like the board. I would probably find the border of my cell, find like the central object, and then do a border to border distance-based measurement. That might be like off the top of my head how I would approach it. But there's probably a couple different ways we could go about it, because we have a lot of channels that we can use here. Like for instance, this channel would looks like it's going to be a really good segment, but because they kind of touch, which is sort of going to be inevitable for what it looks like for the sample type, we'll have to delineate these as individual objects first. But there's a number of ways that we could do that.
Thank you.
Sure. All right.
There's one more question in the chat.
One more question. All right, let's look at that. Can GA3 automatically quantify the subcellular locations of signals, such as distinguishing whether a signal is predominant in the nucleus or the cytoplasm? Yes. The way that I would probably approach that is we would have to define our nucleus area, and then we would have to define our cytoplasm area. From there, I would make two binaries. That would be part of the binary processing, where I would make a binary that's my nucleus, and then I would make a like exclusionary binary that just contains my cytoplasm, and I would do a parent-child relationship. So I would do a parent relationship, parent-child, where the parent is the nucleus and the children are the localization, the signals that are localized in the nucleus. And then I would do another parent-child relationships where the parent is the cytoplasm and the children are localization signals in there. And it would be a children fully contained in parent, and you'd be able to get information on how many different little subcellular localization signals are in each one. And you could measure total like percent of total signal in just the nucleus versus percent in the cytoplasm and get those sorts of measurements. But the way I would think about that is first figuring out my binary and my cytoplasmic binary layers. Sure. Perfect. Again, so I know Anu put in the chat, this stuff is all available at the Live Cell Imaging Core. Please go talk to her if you think you might have some interesting analysis that you want to try. And if you guys have any questions, feel free to reach out to Lauren and I. We're always willing to help. All right. I hope that everybody has a very nice weekend, and we can end it there. All right. Bye, everybody.
Bye. Thank you.